Hello and welcome to my channel. Today we will learn tricarboxylic acid cycle and will understand different aspects regarding tricarboxylic acid cycle. And I assure you that you will clear all your doubts in this particular lecture. Please like, share and subscribe our channel. So let's begin. This cycle was discovered by scientist Sir Hans Krebs. So this cycle is also called as Krebs cycle. First compound produced in this cycle is citric acid. This is a structure of citric acid. This cycle is also called as citric acid cycle. You can notice here that citric acid contains 3 carboxylic acid group. So this cycle is also called as tricarboxylic acid cycle or simply TCA cycle. Now we will understand what is the site of TCA cycle. So it occurs in all organs with mitochondria, means हर एक ऑर्गन जिसके पास माइटोकॉन्ड्रिया है वहां पे टीसीए साइकिल ऑकर होगा. क्लियर? In the mitochondria, it occurs in the mitochondrial matrix. This is an outer membrane of the mitochondria. Then there is an inner membrane of the mitochondria, and inside the inner membrane, this particular part is filled with a fluid called as matrix, and in this matrix, tricarboxylic acid cycle will occur. Clear? There is one very special point regarding enzymes of tricarboxylic acid cycle. All the enzymes of tricarboxylic acid cycle are located or are present in the matrix of mitochondria. This is a matrix of mitochondria, and all enzymes are present in the matrix except one enzyme, that is succinate dehydrogenase. So succinate dehydrogenase is present in the inner mitochondrial membrane. So this is only enzyme which is present on the inner mitochondrial membrane. Rest all enzymes are present in the matrix of mitochondria. So this is a very important point regarding your examination. So please remember it. Now we will take an overview of tricarboxylic acid cycle, and after that we will understand each and every reaction in detail. So this is oxaloacetic acid, which is the first member of tricarboxylic acid cycle, combines with acetyl-CoA and form citrate. Oxaloacetic acid is a 4-carbon compound, acetyl-CoA is a 2-carbon compound, and by combining this carbon they will form a 6-carbon compound which is called a citrate or citric acid. And as the name of the cycle, citric acid cycle, is taken from this particular point. Clear? From citrate, cis-aconitate will be formed. This is also a 6-carbon compound. Then isocitrate will be formed, which is a 6-carbon compound. From isocitrate, oxalosuccinic acid will be formed, which is also a 6-carbon compound. From oxalosuccinic acid, there is a loss of or removal of one CO2, and we get alpha ketoglutarate, which is a 5-carbon compound of the TCA. Again, there is a loss of one CO2 and 4-carbon compound, that is succinyl-CoA, will be formed. From succinyl-CoA, we get succinate, which is also a 4-carbon compound. Then from succinate, we get fumarate, which is also a 4-carbon compound. From fumarate, we get malate, which is also a 4-carbon compound. And from malate, finally we get oxaloacetic acid. So this is a complete tricarboxylic acid cycle. Note some important points out here. This is a cycle, correct, which occurs again and again. And glycolysis is a pathway, correct? So these are all the intermediates of the tricarboxylic acid cycle. First member of the tricarboxylic acid cycle is oxaloacetic acid, and first stable compound is citrate. Please take a note that acetyl-CoA is not a part of tricarboxylic acid cycle. It is not an intermediate of tricarboxylic acid cycle. Clear? Tricarboxylic acid cycle is a common oxidative pathway for all the substrate We will understand about this point later in detail. All the substrate like fat, protein and glucose. When all of these compounds break down, they will finally enter into a common cycle called as tricarboxylic acid cycle. And from tricarboxylic acid cycle, we get NADH plus H plus FADH2, and NADH and H plus and FADH2 will finally enter in the electron transport system from which we get ATP. Clear? So this is a common pathway. We will understand about this particular point later. So let's understand TCA cycle in detail. First of all, oxaloacetic acid will combine with acetyl-CoA and will form citric. They both will combine and form citrate or citric acid. This reaction is catalyzed by an enzyme, citrate synthase. There is a removal of coenzyme A in this step and water is added. This reaction is also called as condensation reaction. This is an irreversible step of TCA cycle. There are 3 irreversible steps in the TCA cycle. This is one of the irreversible steps. We will see next 2 irreversible steps later. This is also a regulatory step of the TCA. This is a structure of citrate. So this cycle is called as citric acid cycle. Now citrate will be converted to cis-aconitate. Citrate is 6 carbon compound and is converted into cis-aconitate, which is also a 6 carbon compound. This reaction is catalyzed by enzyme aconitase. This is an ion containing enzyme and water is removed in this process. Now cis-aconitate will be converted to isocitrate. 6 carbon compound cis-aconitate will be converted to isocitrate, which is also a 6 carbon compound. This reaction is catalyzed by an enzyme aconitase and it involves addition of water. Actually, it is an isomerization reaction. Please note here that conversion of citrate into isocitrate involves 2 steps. So this is a 2 steps process. In the first step, citrate is converted to cis-aconitate and then cis-aconitate will be converted to isocitrate. So in the first reaction, there is removal of water and then in the second reaction, there is addition of water and both the reaction are catalyzed by enzyme aconitase. This is a single reaction having 2 steps and both steps are catalyzed by enzyme aconitase. Now, let's understand some important point regarding aconitase enzyme. So, we know enzyme aconitase contain iron. So, it is iron containing enzyme. It is also called as moonlighting enzyme because it have the enzymatic activity. Also, it regulates the iron in the body. In the next step, isocitrate is converted into oxalosuccinic acid and then oxalosuccinic acid will be converted into alpha-ketoglutarate. Clear? So let's understand important points. So conversion of isocitrate into alpha-ketoglutarate is again a 2 steps process. First, isocitrate is converted into oxalosuccinic acid and then oxalosuccinic acid is converted into alpha-ketoglutarate. In the first step, NAD plus is converted to NADH plus H plus and in the second step, there is a removal of CO2, that is decarboxylation. Dehydrogenation means loss of hydrogen and NAD plus will accept that hydrogen and will be converted to NADH plus H plus and decarboxylation means loss of CO2. Correct? But this decarboxylation is not normal decarboxylation, it is oxidative decarboxylation. Oxidative decarboxylation. Clear? Isocitrate dehydrogenase enzyme is a regulatory enzyme. This reaction is an irreversible reaction, second irreversible reaction, and decarboxylation requires magnesium and manganese ions. Also remember one important point regarding ICDH. Isocitrate dehydrogenase is present in the mitochondria as well as it is present in the cytoplasm of the cell. Mitochondrial ICDH convert NAD plus into NADH plus H plus, whereas cytoplasmic ICDH converts NADP into NADPH plus H plus. Now alpha-ketoglutarate will be converted into succinyl coenzyme A, which is a 4-carbon compound, and this reaction involves decarboxylation. Here, alpha ketoglutarate is a 5-carbon compound will be converted to succinyl-CoA, which is a 4-carbon compound, and this reaction is catalyzed by an enzyme alpha ketoglutarate dehydrogenase. There is a removal of carbon dioxide. So, removal of carbon dioxide, we know it is called as decarboxylation, and as this process also have dehydrogenation, that is NAD plus is converted to NADH plus H plus. So, this decarboxylation is called as oxidative decarboxylation, and this is the second oxidative decarboxylation in the tricarboxylic acid cycle. Clear? We know there are 3 irreversible steps involved in the tricarboxylic acid cycle. We have seen initial 2 irreversible reactions. Now, this is the 3rd irreversible reaction. Clear? Now, succinyl-CoA will be converted to succinate, which is also a 4-carbon compound, and this reaction involves substrate level phosphorylation, that is production of ATP. So, in this reaction, succinyl-CoA, which is a 4-carbon compound, so in this reaction, succinyl coenzyme A, which is a 4-carbon compound, is converted into succinate, which is also a 4-carbon compound. This reaction is catalyzed by an enzyme succinate thiokinase. This reaction involves production of ATP and GTP from ADP and GDP, and there is removal of coenzyme A, and this is called as a substrate level phosphorylation. Clear? There is only one step where substrate level phosphorylation occurs in tricarboxylic acid cycle, and that step is conversion of succinyl-CoA into succinate. Please remember this point. Now, in the next step, succinate is converted into fumarate. Here, FADH2 is produced. In this reaction, succinate, which is a 4-carbon compound, is converted into fumarate, a 4-carbon compound. This reaction is catalyzed by an enzyme succinate dehydrogenase. We know all the enzymes of tricarboxylic acid cycle are located in the matrix of mitochondria, but this succinate dehydrogenase is the only enzyme which is located in the inner mitochondrial membrane. Please remember that particular point. Here, FAD will be converted to FADH2. This is also a dehydrogenation reaction. This reaction is dehydrogenation reaction. This enzyme is present in the complex 2 of electron transport chain. We know electron transport system or electron transport chain is located in the inner mitochondrial matrix, and this succinate dehydrogenase enzyme is also located in the inner mitochondrial matrix. Electron transport system have 5 complexes: complex 1, complex 2, complex 3, complex 4, and 5th complex, that is ATP synthase, which produce the ATP. Out of these all complexes, this enzyme, that is succinate dehydrogenase, is a part of complex 2. We have seen it is not present in the matrix and it is present in the inner mitochondrial membrane. All the other enzymes are present in the mitochondrial matrix, and only enzyme, that is succinate dehydrogenase, is the only enzyme and the TCA attached to inner mitochondrial membrane. Please remember all these important points. Now, from fumarate, malate will be produced, which is also a 4-carbon compound. In this reaction, there is addition of water. Fumarate is a 4-carbon compound converted to malate, which is also a 4-carbon compound. This reaction involves addition of water, and this reaction is catalyzed by an enzyme fumarase. This is a structure of fumarate, and this is a structure of malate. Here, double bond is present in the structure of the fumarate. When you add water, it breaks this double bond, and double bond will be converted to single bond. Correct? So fumarase is a type of lyase enzyme. In the next reaction, malate will be converted to oxaloacetic acid. Malate is a 4-carbon compound, is converted into oxaloacetic acid, which is also a 4-carbon compound. This reaction is catalyzed by an enzyme malate dehydrogenase, and it involves conversion of NAD plus into NADH plus H plus. Clear? So this is a dehydrogenation reaction. Please remember, this is the final step of tricarboxylic acid cycle, and an important point here is that oxaloacetic acid is regenerated with the help of an enzyme called as malate dehydrogenase. Clear? There is one very simple trick to remember tricarboxylic acid cycle, and the trick is Cricket Club of India, OK, send some fund by money order. Cricket stands for citrate, then club stands for cis-aconitate, India stands for isocitrate, O stands for oxalosuccinic acid, K stands for alpha ketoglutarate, SEND stands for succinyl-CoA, SUM stands for succinate, fund stands for fumarate, money stands for malate, and order stands for oxaloacetic acid. So in this way you can easily remember this particular cycle. Now let's discuss energetics and some important points regarding tricarboxylic acid cycle. So first important point is substrate level phosphorylation. So it occurs when succinyl-CoA is converted into succinate. In this step, ADP or GDP will be converted to ATP or GTP. In this step, there is removal of coenzyme A, and this step is catalyzed by an enzyme succinate thiokinase. Please remember, in the tricarboxylic acid cycle, this is the only step where substrate level phosphorylation occurs, and it involves production of ATP. We have two acetyl-CoA molecules with us. For one acetyl-CoA molecule, one ATP is produced, and for two acetyl-CoA molecule, two ATP are produced. So, from one glucose molecule, in TCA cycle, we get 2 ATP from the process of substrate level phosphorylation. Clear? Now we will discuss the energetics. Please remember, for one glucose molecule, Krebs cycle will operate 2 times. Clear? Because from one glucose molecule, we get 2 molecules of acetyl-CoA. For one acetyl-CoA molecule, there is one Krebs cycle, and for another acetyl-CoA molecule, there is again a Krebs cycle. So, for one glucose molecule, Krebs cycle will operate 2 times. Clear? From one Krebs cycle, we get 2 CO2, means 2 CO2 are liberated, that is 2 oxidative decarboxylation will occur. We get 3 NADH plus H plus, 1 molecule of FADH2 and 1 molecule of ATP. Clear? So, from one glucose molecule, we know there are 2 Krebs cycle. This is a data for one Krebs cycle. Multiply it by 2, that is for 2 Krebs cycle, and we know 2 Krebs cycle is equal to for one glucose molecule. From one glucose molecule, we get 4 CO2, 6 NADH plus H plus, 2 molecules of FADH2 and 2 ATP. So, from substrate level phosphorylation, we get 2 ATP. Now you understood? Remember, all the NADH2 and FADH2 produced from glycolysis and tricarboxylic acid cycle will finally enter into electron transport system, and from one molecule of NADH plus H plus, we get 3 molecules of ATP, and from one molecule of FADH2, we get 2 molecules of ATP. Clear? Let's see energy gain after electron transport system for one Krebs cycle, and we know from one Krebs cycle we get 2 CO2, 3 NADH plus 1 FADH2 and 1 ATP. So NADH plus H plus are 3 into 3, because 1 NADH2 will give 3 ATP, 3 into 3 will be giving 9 ATP. FADH2 molecule, from one Krebs cycle we get 1, and 1 FADH2 will give 2 ATP, so 1 into 2 we get total 2 ATP, and we get 1 ATP from one Krebs cycle, which is by substrate level phosphorylation. Clear? Finally, we get 12 ATP from one Krebs cycle. Clear? From one Krebs cycle, we get 12 ATP, and we know for one glucose molecule, Krebs cycle occurs twice. So 1 glucose is equal to 2 Krebs cycle. So 2 into 12 ATP. So total we get from 1 glucose, we get 24 ATP. So this is a complete calculation of the tricarboxylic acid cycle. I hope you understood each and every point. Let's go ahead. Now we will study significance of tricarboxylic acid cycle. Our first significance is, in the tricarboxylic acid cycle, acetyl-CoA is completely oxidized. Let's see detail about it. When oxaloacetic acid combines with acetyl-CoA, it will form citrate. Clear? And from citrate, after successive reactions, succinyl-CoA will be formed, and again from succinyl-CoA, oxaloacetic acid is again regenerated. Clear? So This is a first half and this half is a second half. And the first half, there is an oxidation of the acetyl-CoA, and in the second half, there is a regeneration of oxaloacetic acid. Acetyl-CoA contain 2 carbon atoms, and here these 2 carbon atoms are lost as CO2 in the first half of TCA cycle. So this phase is called as oxidation of acetyl-CoA. Clear? Our second significance is that it is truly an amphibolic pathway. Amphibolic pathway means catabolism plus anabolism. Any pathway having both the processes, that is catabolism plus anabolism, that particular process or pathway is called as amphibolic pathway. Clear? We know catabolism means breakdown and anabolism means to synthesize. So catabolic role of TCA cycle is to break down acetyl-CoA. So acetyl-CoA is completely oxidized, we know, and 2 carbons of acetyl-CoA are liberated or released in the form of CO2. Clear? And what is an anabolic role of TCA? Let's understand. Different intermediates of TCA are used to synthesize some molecules. Correct? So citrate can be used to form fatty acids. Alpha ketoglutarate can be used to form glutamate, and from glutamate we get neurotransmitter, that is GABA. Succinyl-CoA can be used as a substrate for the heme synthesis. Oxaloacetic acid can be used for the formation of glucose. Clear? So these are the intermediates which are used to form some molecules. Clear? So here we are making or synthesizing some molecules from these intermediates. This is called as an anabolic role of TCA. Clear? TCA cycle have both catabolic role and anabolic role. So this is truly an amphibolic pathway. Our third significance is it involves anaplerotic reaction. Clear? Let's understand what is meant by anaplerotic reaction. Anaplerotic reaction, in simple words, they are also called as filling up reaction. Means in the anabolic role, we know we are using different intermediates of TCA cycle to form some other molecules. We are using citrate, alpha ketoglutarate, succinyl-CoA and oxaloacetic acid to form fatty acid, glucose, GABA or heme synthesis. When you do that, quantity of citrate, alpha ketoglutarate, succinyl-CoA and oxaloacetic acid will deplete. Clear? इसका quantity कम हो जाएगा. So for next coming acetyl-CoA molecules, we don't have these intermediates. We don't have oxaloacetic acid to receive the acetyl-CoA molecule. To fill this used oxaloacetic acid or to fill this used citrate, to fill this used alpha ketoglutarate, to fill this used succinyl-CoA, there are some reactions, and that reactions are called as anaplerotic reactions. So what is the function of anaplerotic reaction? जो भी oxaloacetic acid, citrate, alpha ketoglutarate and succinyl-CoA यूज हुआ है कुछ मॉलिक्यूल्स बनाने के लिए, तो उसको फिल अप करना, अगेन उसका जो क्वांटिटी है वो नॉर्मल पोजीशन पे लेके आना. सो दोस रिएक्शन आर कॉल्ड एज anaplerotic reaction. We will not go in detail about the anaplerotic reaction. This is just an idea about anaplerotic reaction. Clear to you? TCA cycle is also called as final common oxidative pathway for some macronutrients like carbohydrates, lipids or fats and proteins. Let's understand why it is called as final common oxidative pathway for all the micronutrients, because we know we have fat, it's also called as lipid, we have carbohydrate, we have substrate as protein. Fat will break down into fatty acid and glycerol. Now fatty acid and glycerol again break down into acetyl-CoA by a pathway called as beta oxidation. Carbohydrate will break into pyruvate, proteins will break into amino acid, again the pyruvate and amino acid will break down and form acetyl-CoA. Whether substrate is fat or carbohydrate or protein, they will initially break down by different pathways and finally form acetyl-CoA. which is a common intermediate of all the three substrates, and this acetyl-CoA then will enter into the tricarboxylic acid cycle and gives NADH plus H plus and FADH2, and finally NADH and FADH2 will enter in electron transport system and will generate ATP, and this ATP are used by the body. So tricarboxylic acid cycle is a common oxidative pathway for fat, carbohydrate and proteins. Clear? And in TCA we know acetyl-CoA is completely oxidized and this NADH plus H plus and FADH2 called as reducing equivalents are generated. Clear? So this is a final common oxidative pathway. Clear? Please remember this point. Please remember one important point, and that is NADH plus H plus and FADH2 are the reducing equivalents or reduced coenzymes. NADH and H plus is formed in both glycolysis and TCA cycle, but FADH2 is only formed in TCA cycle. Clear? This is one important point, and that is one NADH plus H plus gives 3 ATP, we know, and one FADH2 gives 2 ATP. Please listen carefully. Students of 12th standard who are appearing for NEET or AIIMS, please follow this criteria, that is one NADH plus H plus gives 3 ATP and one FADH2 gives 2 ATP. And students who are doing their graduation, please follow that one NADH plus H plus will give 2.5 ATP and one FADH2 will give 1.5 ATP. Clear? So entire calculation will be changed. We have seen calculation according to the 12th standard. Okay? I hope this lecture will help to understand the points regarding TCA cycle. Please like, share and subscribe our channel. Learn with No Easy. Thank you.